Okay, so for A-level biology, where we're going to start is we're going to start with monomers and polymers. So we're going to start with biological molecules, how they're formed, and the basis of them, because basically these things get revisited a lot throughout the course. And what ends up happening is further on in the course, you get questions that might be to do with the circulatory system, or you might get questions to do with plants in particular, about their cell walls and how the xylem and phloem work, and then they start relating them back to biological molecules such as glucose and cellulose and starch. And so learning about them separately first is really good and really useful, because what that allows us to do is that allows us to sort of understand the chemistry behind them, and work out all the different parts of them before sort of confusing them then with other information. So then when we revisit it later on, we go, oh yeah, I remember that because I did it back at the beginning. So that's sort of a basic idea of where we start from. So yeah, monomers and polymers. And it is a very chemistry based first topic, but it is quite a nice topic. So monomers and polymers. Let's get this working. Why are we not skipping slides? So monomers and polymers. So we'll start off with monomers. Monomers are something you might have come across before. You will have done monomers before, but sometimes we don't use the word monomers. Sometimes we use like a simple molecule or a small molecule. So for example, when we've spoken in the past, when you've had GCSE and you've done about digestion, and there'll be conversations about, oh, the carbohydrates are broken down into glucose, or the fats are broken down into lipids, or this is broken down into that. When we're breaking these things down, we're basically breaking polymers into monomers. And officially, a monomer is a single subunit. And what we mean by that is a monomer is as small as we can get. So if we have a look here, this is a single monomer here. So a single amino acid would be a monomer. A single molecule of glucose. So obviously we could break them down smaller to the atomic point, but in terms of biology, it's a single subunit. This is, for it to be what it is, this is as small as we can get it, and a polymer is lots of them joined together. So a monomer is just one on its own, and a polymer would just be lots of monomers joined together. So our main examples are things like amino acids are monomers, monosaccharides, fructose, and fructose. So yeah, these come up a lot. And what we start to do is we start to talk about monomers and then building them into polymers. There's not much talk of just having them on their own. When we do have them on their own, it's when we're actually using them. We don't really store any molecules as monomers often in our bodies, just because it's not that useful. So yeah, we've got amino acids, monosaccharides, and fructose. So monomers can be joined together, like we've got this image down here, and they can make a polymer. That is what we're going to look at now. So polymers, I have lost what I wanted to come up, is a complex molecule. We say it's complex because basically it's more than just a single subunit. Monomers, we would say, is a simple molecule. Polymers are a complex molecule. They're made up of many monomers. So for example, like we said with monomers are amino acids. So for example, the polymer version of amino acids would be proteins. Obviously then DNA is a form of a polymer, starch is a polymer, enzymes are polymers, and we have loads and loads of different polymers. But they're these complex molecules. They're just made up of lots and lots of different polymers. How they join is they join using covalent bonds. So these bonds here would be a covalent bond. So we've got a covalent bond being formed here. Oops, it's gone a bit off to the side, but never mind. So that would be a covalent bond being formed there. Now we'll look at how that covalent bond is formed in a second, but that's just the same as when we think back to GCSE in chemistry. And covalent bonds is that sharing of electrons. We're going to have some sharing going on. So Condensation reactions. Condensation and hydrolysis reactions are just going to come up loads and loads in this topic. They come up loads in biology, they come up loads in chemistry. So the quicker and sooner you're happy and confident with hydrolysis and condensation reactions, the better it is. So let's have a quick look here. Condensation reactions. So basically, condensation reactions is when we want to join two single subunits together. So a fantastic example of this would be as joining some monomers together. We want to join some monomers together, so we are going to use, I'm going to give up trying to write with that because that is not working. No, we will stop. If we want to join some monomers together, we would use a condensation reaction. Condensation reaction, and I've got something coming up over the top of that now, of course, I don't know, is where we get point eruptions. It's the formation of larger biological molecules. So, for example, we've just spoken about monomers. We want to make a polymer. We're going to use a condensation reaction. Condensation reaction is basically just a reaction where water is released. So if you think of condensation being formation of water, water vapor from a gas to a liquid, we think about water being released here. So if we look at this molecule, for example, we've got here our water molecule here. We've got the OH and the H together. What happens is this OH and H is lost. They go off together and form a water molecule, and then we get this other bonding going on down here. This bonding then joins the two monomers together to make a polymer. What we call this bonding is a peptide bond. And so we'll talk about peptide bonds in a bit more detail in a bit, but this joining here together makes a peptide bond. So we have our H2O being formed over there, and we get our peptide bond being formed. So that's a condensation reaction. Condensation reactions are going to take place when we are making bonds for things like polymers. Opposite of a condensation reaction is a hydrolysis reaction. So, for example, I've got an example of hydrolysis of esters. Basically, hydrolysis and condensation reactions are the exact opposite of each other. Hydrolysis reactions, you're going to break down your large molecules. So, for example, as we're talking about here, we're going to break down our polymers. And we're going to break down our polymers into smaller molecules, and we are going to, and that is going to require water. So as you can see here, if I can find my mouse, we have water starting to appear. So what has to happen is we have our original ester molecule here. Water is added, and with the formation of water, what that does is it allows those bonds between them to break. So those peptide bonds that we have back in our condensation reaction that are formed, we add water in, we hydrolyze the molecule, this bond here breaks, this peptide bond breaks, and that means that we've then got two separate molecules, so we've got two monomers. And that's generally the idea of hydrolysis reactions and condensation reactions. Condensation reactions, we're going to build a larger molecule. Hydrolysis reactions, we're going to break them down. Both involve water. One is adding water, and one is taking the water away. So I'm going to start off looking at carbohydrates. I'm going to look at monosaccharides and disaccharides. So carbohydrate group is the first group that we look at in biology at A level. So we're going to look at how carbohydrates are constructed. We're going to describe the structure of monosaccharides. So we have two structures of glucose that we have to learn. Now you have to learn it off by heart and know the difference between them. You can quite regularly get asked in an exam to draw alpha glucose or get asked to draw beta glucose. So it's really important that that is something that is memorized and learned. We're then going to look at how monosaccharides are linked together to form disaccharides. So adding some of that information we've just got from monomers and polymers, and then look at alpha glucose forming maltose and starch. So carbohydrates. Most carbohydrates are polymers. They are not generally monomers. We have a couple that are monomers, but most carbohydrates are polymers. So they are lots of monomer units that can join together through condensation reactions. They are large, they are complex, and they are long chains. So for example, like we said before, some examples of carbohydrates, we've got nucleotides, we've got amino acids, we've got monosaccharides, are examples of our polymers here. They are made from monosaccharides. So monosaccharides, we've got the prefix of mono here meaning one, di meaning two, tri meaning three. So monosaccharide is like a monomer, a singular. We've got a monosaccharide such as fructose or glucose. They join together to make a more complex carbohydrate. So we'll have come across glucose loads when you've done GCSE biology, and it's been spoken about. Eat carbohydrates and the carbohydrates get broken down into glucose. Obviously, there's more than one sugar that carbohydrates can be broken down into, but we often use glucose as the example. We generally use glucose as the example because glucose is used very regularly in respiration. So it's sort of a molecule that we like to use a lot to talk about because we can sort of link it to many places. But we have more than one monosaccharide, which is the important part. So what do we know about monosaccharides? They are a single sugar unit. So they are a monomer. They are sweet and soluble. They contain carbon, hydrogen, and oxygen. And they are classified according to the number of carbons a molecule has. So a single sugar unit just means that it's a monomer. And they've got carbon, hydrogen, and oxygen inside of them. And what we mean by being classified to the number of carbons a molecule has is, I will show you an example of one here. So this is our glucose, and we would count how many carbons are inside this molecule of glucose. We've got 1, 2, 3, 4, 5. So we've got a pentose sugar. So any sugar that has 5 lots of carbon in it would make it a pentose sugar. You can have a tri sugar and a di sugar. So what are the main biological roles of glucose in organisms? We're going to sort of basically go back to glucose. Glucose is the main one that we talk about. It's the main one that's brought up at GCSE because it's one of the main ones involved in our bodies. So glucose is an instant source of energy. It's used in aerobic respiration. Why is that really, really important? So glucose is our instant source of energy, and the idea of glucose is that we can use that energy straight away. We use it in aerobic respiration. We also can use it to make other substances such as sucrose, starch, cellulose, and glycogen. Glucose is generally, we break polysaccharides and disaccharides down into glucose because glucose isn't great for storage. Because it's this instant source of energy, the problem is it will get processed and it will go through reactions constantly. If we just had glucose going around in our body, it's soluble. It's not a useful molecule for storage. So it's why we go through this process with carbohydrates of having complex polymers, complex mono, complex di and polysaccharides, and breaking them down into monosaccharides. It's because actually the monosaccharides are this fantastic instant source of energy that we can use, for example, in aerobic respiration, but they're really, really rubbish for storing. They're soluble, so they won't store in our body, so we have to store them in another form. So these are the two glucose molecules you have to know about. So both these molecules are glucose. They both have exactly the same molecular formula, which is C6H12O6. Doesn't matter if it's alpha glucose or beta glucose, they both have exactly the same molecular formula. These are called isomers when they have a different arrangement. So if we have a look here on our alpha glucose, what has happened is we have the carboxyl group has been swapped round in the directions here to here. I know one has got all the C's drawn on and one hasn't. This is something that we'll look at more in chemistry, but wherever you get to a point on a display formula, there would be a C there, and we don't necessarily always draw them out. So this has had a switch round. So alpha glucose, the H is at the top, beta glucose, the H is at the bottom. It's only on this fifth carbon. So we count the carbons from here. So we go 1, 2, 3, 4, 5. On this fifth carbon, we have the carboxyl groups switched around in directions. That's all you need to know about alpha and beta glucose for now. Is it isomers? They have slightly different arrangements. So it's the same molecular formula, but those two are switched around. You have to know this off by heart. It's one of those ones that people sort of try and come up with really strange ways of remembering them. Some people go, okay, A comes first in the alphabet and H comes first in the alphabet before O. So alpha glucose has H at the top. Beta glucose, B comes second, and that has H at the bottom. Not saying that's really a fantastic way of remembering it. Lots of people just learn it off by heart and go, oh yeah, this way and that way. But people do have different ways of remembering it. I probably, when I learned it originally, went with that sort of alpha begins with A, H is before O, so it'll be that way around. It's not necessarily the clearest way, but it is a good way of remembering it. So that is two different forms of glucose, and you do have to know these off by heart, drawn out fully. And you have to know the difference on this fifth carbon of the hydroxyl group, which way round they are. These are hexose sugars because we've got 1, 2, 3, 4, 5, 6 carbons on them, and they are displayed out separately. So that is sort of one that you just want to print off, learn, and copy down loads and loads of times to remember. There's not really much more to that at the moment. So monomers, monosaccharides, we have loads of them. We have two. We want to form the disaccharide called maltose. What we do is we have two glucose molecules. Two glucose molecules can join together to make maltose. Maltose is a disaccharide. Here we have the prefix di, meaning two. I don't know what's happening anymore to my mouse. Here we go. We've got disaccharide, di meaning two, and we are going to join them together. If we think back to when we talked about joining monomers together to make polymers, we said, oh yeah, you use a condensation reaction to join two monomers together to make a polymer. Exactly the same. To join two monosaccharides together to make a disaccharide, condensation reaction happens. So again, you can see here you have the OH and the H from the two separate glucose molecules. If you look at the glucose molecules here, you have got two copies of alpha glucose. The OH and the H join together to make H2O. The H2O is removed and we get this bonding happening here. We call this bond, and this is the important bit that we have to remember, a 1,4 glycosidic bond. And what we'll do is we'll go back and we're going to number on here our carbons. So call it a 1,4 glycosidic bond because we number our carbons on our hexose sugar. So for example, this one would be carbon 1. This one is 2, 3, 4, 5, oops, and there's number 6. Same thing will happen on this one. 1, 2, 3, 4, 5, and 6. So when we have maltose forming, we call it a 1,4 glycosidic bond because the first carbon here and at the 4th carbon, this linkage is made, and we call it a linkage, and it's a glycosidic bond. And that is formed between 2 monosaccharides, and we call it a 1,4 glycosidic bond. And we call it a disaccharide when we have 2 monomers joined together. So this is a condensation reaction forming a 1,4 glycosidic bond. So, quick summary of that again. When we're forming the disaccharide maltose, we use 2 alpha glucose molecules, and the carbon 1 and carbon 4 meet. The hydroxyl group, the OH group from carbon 1, and the hydrogen from carbon 4 react together. Water is expelled, so we remove the water, and we get this condensation reaction being formed. We can reverse this just by adding water. So if we add water to maltose, hydrolysis reaction will happen. It will break at the 1,4 glycosidic bond, and then we'll have 2 molecules of glucose being present. So these are all reversible reactions, and we officially call it an alpha 1,4 glycosidic bond because it's formed from 2 alpha glucose molecules. Don't know why I've got a little A appearing there. Let's go here. So polysaccharides. Monosaccharides, monomers, 1 subunit. Disaccharides, 2 subunits. We're getting towards, they are technically a polymer, but it's just 2 monosaccharides joined together. Polysaccharides. These are polymers with subunits of many monosaccharides. They are repeated condensation reactions happening again and again. So to join them, we have a condensation reaction, and a condensation reaction. Normally, for a polysaccharide, we have got thousands and thousands of monomers joined together. And polysaccharides are not sugars. And that's a really, really important fact. Polysaccharides are not sugars. So polysaccharides can be things such as starch, cellulose, and glycogen. I'm going to have a little look at starch, cellulose, and glycogen, about the differences for them, what we use them for, and why we have them in our bodies and why plants have them. We will also have a quick talk about testing for sugars. This is exactly the same as at GCSE for reducing sugars, but we're adding something called non-reducing sugars as well. So starch, we're going to look at starch. As you probably remember, starch, right back at GCSE, is a polysaccharide, and starch we test for using iodine. So starch is a polymer of glucose, and it's a plant storage polysaccharide. And what we mean by a plant storage polysaccharide is this is a storage molecule. It is a molecule that stores glucose, and it's a way that we're basically storing the glucose up. We've already discussed that glucose is soluble. It's not a good way to store up energy to be used at a later date. Glucose is to be used now, straight away. It's almost like having a Mars bar that's going to give you the energy straight away, but we're not going to save any. Starch is a storage molecule. So starch is a plant storage molecule. We don't use it in our cells as humans, and it's made up of 2 different substances. We have amylose and amylopectin. So amylose. Amylose is made up of condensation reactions between alpha glucose at the 1,4 links. So exactly the same as when we were talking about forming maltose, but amylose is a very, very long molecule. So 1,4 links again. Amylose has thousands of condensation reactions formed in, and what ends up happening is we get coiled springs being formed. So why should a storage polymer be large and coiled? Well, we want a storage polymer to be large because if we try to store energy in the form of our disaccharides, maltose, we're not really storing much in the space. So we basically want to cramp in as much as possible into the smallest place possible. So we want to have lots stored up. So we want our storage molecule to be large. We want it to be long because otherwise we're joining all these monomers together for no good reason, because we're just going to break them down again really quickly. So we want them to be large. And coiled is really good because it saves space. So here we go. This is a spiral structure of amylose, and it is a long unbranched chain. And what we mean by unbranched is we don't have any extra bits coming off it around here. It is just a long unbranched chain. And that's really, really good. The length of it, and this length is really, really helpful because we are just cramming in as much as possible. Because it doesn't have any branches, we are able to make it into coils and spirals and really squeeze it together. So what happens is those alpha 1,4 glycosidic bonds, these basically, the angles of them allow this coil shape to be made. And we'll look at the shapes and the angles of bonding as well when we look at things like proteins. And all to do with our bonds and our electrostatic charges basically means that when we're making polymers, that they all fold in specific shapes. And so this 1,4 glycosidic bond allows this coil shape to arrive. This coil shape is fantastic because it's really, really compact and it's really good for storage. If you think like the small intestine and how long that would be if you took it out your body, amylose works in that same sort of concept. We can put a lot into a small space because we can coil it and squeeze it in. We haven't just got this really, really long chain that would take up loads of space. We can coil it and squeeze it all together. So amylose is really, really good like that. So it's spiral structure and it's part of the starch. Obviously, have the other part of starch, which is amylopectin. Amylopectin is a long branched chain. So it is a condensation reaction between alpha glucose again at those 1,4 links again. So it's going to make alpha glucose 1,4 glycosidic bonds. But we can also get branches of 1 to 6 links. What this does, and I'll show you this on the next image, is it makes a different shape ever so slightly. So we get this. So this is the amylopectin, and here's one of our side branches. Now, what's really, really good about side branches is it allows enzymes to get in. So if you think, let me draw on here. If we just draw a coil which was coiled up, and it would coil in like this, it's sort of quite hard if we had an enzyme coming along to try and get in there to start to break it down. Really hard for an enzyme to get in. So these side branches, these branches on amylopectin are really, really useful because what they do is they allow enzymes to actually get into it. Because if you think we've got a chain, but we've got these side chains coming off. So these side chains stop the molecule from coiling as tightly together as amylose. So amylose is going to be really super coiled. Amylopectin is still going to be coiled, but it's got these few more gaps in it because of these side branches. So these side branches allow our enzymes to get in, and these enzymes are going to come in and they're going to break down those glycosidic bonds. Why is that important? Well, amylopectin is part of starch. Starch is a storage molecule. The main goal of starch is for us to break down the molecule back into glucose. So that glucose can be used in things like respiration. It is a source of energy. So we want enzymes to be able to easily access those 1,4 glycosidic bonds to break that polymer down back into a monomer. So our glucose can be released easily. So these side branches are really, really good and really, really useful for releasing our energy in that way. Another good thing about amylopectin and about starch in general is it is insoluble in water. So it doesn't cause any osmosis to happen. So it will not affect the water potential of the plant, and or any of the cells that it's taking place in. So amylopectin and amylose and starch are all insoluble in water, which is really, really important. If they were soluble in water, it would cause an osmotic effect. If this osmotic effect was created, it would mean that we'd have movement of water in and out. It would mean that we would have movement of the glucose, so the storage molecule wouldn't stay in the same place. We don't really want a storage molecule to be wandering around our bodies, or a plant's body, for example. We want them to be stored in the same place, so the enzymes can always go to the same place to break it down. So being insoluble in water is also really, really useful. So we have those 1,4 links form a helical structure, and a helical structure is very similar to coiled, but it has just these little breaks in that we talked about to let them enzymes in. So again, this is that amylopectin. They're mostly 1,4 links. We have a couple of 1,6 links, and we have those nice little gaps so that coming in, we can get these enzymes to come in and start to break it down. Then we have glycogen. Glycogen is fantastic. It is the main storage polysaccharide in animals, so it is our equivalent to starch. It again is an energy form from glucose. It's lots of glucose molecules joined together until we eventually get glycogen. And it is a polysaccharide of alpha glucose again. Starch is a polysaccharide of alpha glucose, and so is glycogen. It is very, very similar to amylopectin. So not only is it our equivalent to starch, its actual structure and forms are very, very similar to amylopectin. But glycogen has more side branches. And we'll talk about why that is useful in a second. So glycogen is our storage molecule. We find a lot of glycogen in our liver and our muscle cells, where a store of energy is really needed. So we store our energy close to the source of where energy is going to be used and needed the most. So for example, muscle cells are going to need a really quick response of energy to be provided for them to be able to contract, for them to move. There's no point storing glycogen in our nose, for example, because then it would have to travel all the way to our arm and our biceps of a muscle for it to move. So we store where we need it. It's to make it a really highly efficient system. Bit of a side point, fungi also store glycogen. So whilst we say, oh yeah, glycogen is the animal version of starch, fungi also do have glycogen. It is, like we said, very similar to the structure of starch. Actually, rather than just saying starch in general, it's very, very similar to the structure of amylopectin. So amylopectin. But it is more branched. Why is being more branched even better? Well, we said amylopectin is branched so that the enzymes can get in to break down the molecules so that we have glucose re-released. Plants are a bit more reliable and less likely to need a sudden burst of energy. Humans, on the other hand, and animals, because we move and how we move and how our metabolic processes work, is we're not as constant. We often will need big amounts of energy suddenly and not as much for longer periods of time. So having extra branches is better, because the more branches we have, the quicker the glucose can be released, because those enzymes can get in easier to break down that molecule of glycogen. So it's all about the speed and the efficiency of breaking down glycogen. Plants don't need that because they don't need that sudden burst like we do, so they don't have to have as many branches. We have more branches because it's a little bit better for us, because we can get that energy quicker. Glycogen will form tiny granules inside your cells, and these are usually associated with the smooth endoplasmic reticulum inside our cells, which is something that we'll look into more. But our smooth ER is areas where we have lots of respiration and other metabolic reactions taking place. It is compact. It's really, really compact storage, like starch, so it's fantastic for that. And then finally, oh, no, sorry. Each glycogen molecule also contains a few thousand glucose units. So it's huge, but it's compact. So it's great. So this is glycogen in the storage polysaccharide animals. So we have this beautiful spiral shape being formed. So you can see really easily how all the enzymes can come in and start to break down the molecules really quite easily because we've got this coil being kept open. So this here is showing us where our polymer branch would be. This is a branch coming up off glycogen. And so we have these branches being formed and they're really useful to help that enzyme get in and break them down. So we have our 1,4 links like we've spoken about, but we also have those 1,6 links. And these are the links that form our branches. And finally, the third polysaccharide we're going to look at is cellulose. Cellulose is another plant polysaccharide, and it is present in plant cell walls. Cellulose is fab because it has a really, really slow decomposition. Something like starch is a lot quicker to decompose, but this is really, really slow. It is the most abundant organic molecule on the planet, and it is strong. In these situations, generally in plants, cellulose isn't used as a storage molecule for energy. It's why having a slow decomposition is really useful. We don't actually want this to be broken down. We don't actually want to turn this back into glucose. Plants are just using it for its ability to be really, really strong. It's why we find it in plant cell walls. It is a polymer of beta glucose. So this is the first time that we've sort of come across beta glucose since we've been talking about the two different types of glucose. And we're going to have a look at how it forms together. So it's polymer of beta glucose, and it has a beta glycosidic 1,4 bonds. So it's exactly the same 1,4 bond as we would have in the alpha glucose, but it's on the beta glucose, so we call it beta 1,4 links to make up this polysaccharide. So if carbon 1 and carbon 4 are to react, one glucose molecule actually has to flip through 180. And I'm going to show you in a second what that looks like. Because it has to flip, this is what makes cellulose really, really strong. And this structure has hydrogen bonds that hold it together. So these hydrogen bonds hold it together, and it's composed, and what we can use it for as well is not only for making the plant cell walls, but in there we have something called a microfibril. And these microfibrils are basically these really strong structural supports that are almost like, I don't want to use the word bone, but they almost look, when you look under a microscope, like thin pieces of string or bone, and they basically work like a skeleton. And there's these really strong, fine pieces of structure which make a structural support. So cellulose, we don't want it to break down because we want to use it for that. So why is it important that cellulose is made of beta glucose? Well, it's important that cellulose is made of beta glucose because for that carbon 1 and carbon 4 to react, one of those glucose molecules needs to flip 180 degrees. That flipping 180 degrees is what makes the cellulose so, so strong. And let's have another look at cellulose. So here's our cell walls, and this is what I'm talking about when we say microfibrils. These microfibrils look almost like pieces of cotton, and these are what are polymers of beta glucose. So if we look, we have a glucose monomer here. We join them together and we start to get polymers of beta glucose, and we get these cellulose chains. Lots of cellulose chains together make a microfibril. Lots of microfibrils together make a fibril. And then these are found in our cell walls. So we have 70 chains of beta glucose combined to form a microfibril, and lots of microfibrils are held together to form fibers. So we just basically, it's almost like the idea of we have cells, lots of cells together make a tissue, lots of tissues make an organ, lots of organs make an organ system, lots of organ systems make an organism. It's that same concept with building blocks, building it up to make it more complex. So what we're going to have a quick flip to now is lipids. So the last little bit we're going to look at is lipids. What you will start to notice is that lipids and carbohydrates, whilst they are different molecules, they start to have lots of similarities. The way we form the monomers into polymers, we're still going to be using our condensation and hydrolysis reactions. So we're going to look at triglycerides, how to form them, and the roles of lipids in the body. So we have two main lipids that you need to know about. We have triglycerides and we have phospholipids, and they're the only two that you need to know about. So we're going to look at what they're made up of and what their different functions are. So the first one we're going to look at are triglycerides. Triglycerides are made up of just two different molecules, and they're made up of a glycerol and 3 fatty acids. So again, we have that prefix giving us a clue. So we have prefix tri involved here. So tri meaning 3. So triglycerides, we have 3 fatty acids to 1 glycerol. And here is a massive surprise. Fatty acids are our monomers, and we also have our glycerol. How do we join 2 monomers together? A condensation reaction. However, to make 1 triglyceride, we have 3 condensation reactions take place. Because if we look carefully, we are having a bond being formed here between this fatty acid and glycerol, a bond being formed here between this fatty acid and glycerol, and the same here. So when a triglyceride is formed, 3 molecules of water is produced during this condensation reaction. Triglycerides, they are insoluble in water, and this is a really important part of them. So when we join them together and we make these bonds, when we talked about these bonds were called glycosidic bonds back with carbohydrates, they have a completely different name when we talk about in lipids. So these condensation reactions create these bonds between the glycerol and the fatty acids. These bonds are called an ester bond. All you need to know right now is it's called an ester bond. At some other point in chemistry, we'll actually look into what an ester bond is, how they work, but for now, we call them an ester bond. So what happens is, here is our glycerol molecule here, and here are our 3 fatty acid molecules. There are the water molecules that are going to join together, they're going to leave, and we're going to have that condensation reaction happening so that we can form a triglyceride. So we have 3 ester bonds forming. That oxygen from the glycerol, when it joins up with the carbon from the fatty acids, we have something being formed that is an ester bond. And so like we said before, we have 3 condensation reactions happening, so therefore we have 3 ester bonds being formed. Triglycerides, they are made up of hydrocarbons, and I just want to draw this on really quickly. So, oops, let's get the pen. We have oxygen, we have a hydroxyl group, we have a double bond carbon, we have a single bond carbon, and we have R. R groups, which we'll come across loads as we carry on, they are basically groups that can change. So they can be different ones depending on what the hydrocarbon is. So we don't need to really worry about that. So these are interchangeable groups. So this is our fatty acid basic structure. The R group can change. It can be many variations of many different atoms. R group stays the same. R group changes, sorry, and everything else stays the same. They are hydrophobic. Hydrophobic means, well, hydro meaning water, phobic meaning scared of. The hydrophobic molecules means that if you don't like water, you can have hydrophilic, which means water loving. The hydrophobic molecules is what makes them insoluble. They do not like water. You can almost use the word like. I know it doesn't technically like or dislike it, but we would call them water hating molecules. So they are insoluble in water. And in a moment, we will have a look at what that forms. And because of the different bonds and the different charges of them, it creates this hydrophobic area. So we will look into that in a second. So triglycerides, why do we have them? Again, like glycogen, they are an energy storage molecule. They have long hydrocarbon tails and they contain lots and lots of chemical energy. This chemical energy is released when the triglyceride is broken down. So if we broke this triglyceride down, if we went through a hydrolysis reaction and broke those 3 ester bonds, we are releasing energy when that happens. They have 2 times more energy per gram than carbohydrates. It's why we store a lot of our energy as fat. It's a lot better than carbohydrates because energy per gram is double. They're insoluble. They're insoluble in water, which means they do not affect water potential where they are found. They form something called a micelle, and I have not put a micelle, but it's annoying. So I'm going to draw you a micelle really quickly. A micelle is basically, because I said that they were hydrophobic, actually I'm going to get a picture of one really, really quickly. Let's escape this. Do you want to keep in annotations? What's that? Let me get an image of a micelle really quickly. Sorry. So let me get a micelle. This is basically, if you think about when you put oil in water and you get them like little globules being formed, this is what a micelle is. It's those little globules that are formed during when water goes into, oil goes into water even. Shouldn't try and talk and do something at the same time because I struggle. So I want to share the screen again. What I've done. Share screen. Share. Okay. Don't tell me I've done all that. It's not going to work. Ah, here we go. So, got a picture of a micelle here. So a micelle is basically, we said that they have hydrocarbon tail, which is hydrophobic. So basically what happens is these hydrocarbon tails, the fatty acid tails, are hydrophobic, so they don't want to be towards the water. The glycerol heads are hydrophilic, so they're happy to go out towards water, and all the tails form inside. So they basically make this big circular molecule together where all the tails are inward, so water cannot penetrate inside. It becomes more apparent when we start talking about phospholipids because they make this really cool bilayer, and that is basically what the whole of the outside of our cell membranes look like. But yeah, we make these micelles and bilayers. So saturated or unsaturated fats. Unsaturated fatty acids have double bonds, so they have carbon-carbon double bonds. They have a really low melting point, and so they tend to be liquid at normal temperatures. Saturated fats have no double bonds, and they tend to be solid at normal temperatures, and that's the main difference between them. We call them saturated because they are saturated with an extra hydrogen. So where the carbon-carbon double bond is, these have been saturated with an extra hydrogen. So we're saying they're saturated, they have an extra hydrogen that takes its place. Unsaturated fats have at least one double bond between carbon. For example, this one's got 1, 2, 3 in there. It could have 3, it could have 4, etc. But it's got to have at least one in there. And it creates a kink in the chain. And because of this kink in the chain, they have a really low melting point. This kink in the chain is what creates this low melting point. So it's what causes them to be liquid at normal temperatures, meaning room temperatures. Because saturated fats do not have that kink in the chain, they are solid at room temperature. so they have a higher melting point. So what is the role of triglyceride in our body? It is a storage of energy. It's insoluble. It's really good, but it's insoluble because that means that it's better at storing. It's made up of fatty acid chains. So the last bit are phospholipids. Phospholipids are structurally different to triglycerides only by a small bit, and they have different biological functions. Instead of a triglyceride, which has 3 fatty acids to a glycerol, these have 2 fatty acids attached to a glycerol, but they also have an inorganic phosphate group that is attached to the glycerol in place of that 3rd fatty acid chain. So what you sometimes see them drawn as is they are sometimes drawn as this. They are sometimes drawn as we've got our glycerol, and this is really badly drawn. We've got a fatty acid coming off, a fatty acid coming off, and then we have a phosphate group here. My pen has broken, so we've got my dodgy drawing instead. So they have this inorganic phosphate group attached to them in place. What is great about these are found in our cell membrane, and one of our fatty acids has been replaced by a phosphate group. And what these do is they form a double layer, which I think I have an image of. Oh, didn't want that to stay like that. This is a double layer that I was talking about. So triglycerides form that micelle, which is basically like a cell. It's circular and it's spherical. Phospholipids form a bilayer where you literally just have one layer of phospholipids on top of another layer. And that is because they have a phosphate head and a fatty acid tail. The fatty acid tail is hydrophobic. The phosphate head is hydrophilic. So what happens is this creates 2 defined layers between them. So rather than just having loads and loads of tails just meeting in together in the middle, we have a defined layer of the side that likes to fill water and the side that doesn't. This means that we're not just going to have water coming in and out of our cells all the time due to osmosis because of this insoluble layer in the middle. It's why we have to use things like osmosis for water to come through. It's why we have actual structures taking place. So the center of the bilayer is hydrophobic. And then last little bit is just testing for lipids. This is just a recap from what you've done at GCSE. And we call it the emulsion test. So we test for lipids. If it is a solid food, so I'll add this bit on, we would break the food into really, really small pieces, shake it up with alcohol, so ethanol, in a test tube, and any lipid present dissolves in the alcohol. What we would then do is we would pour the ethanol, the alcohol, into a clean test tube, and we would add an equal quantity of water and would shake this really, really vigorously. If there is a lipid present, if there is fat present, a milky white emulsion is formed. If there's no lipid present, the mixture stays clear. This is the official method that we would have used at GCSE. We often don't do it in school this way because it often doesn't work, and we often don't have solids, and we have things like cheese and we just rub them and see that they've got fats on. This is officially what we would do. We need to break them up, put ethanol into them, pour the ethanol off into a clean test tube, mix it with water, and we should get this milky emulsion being formed. And that is once I stopped recording.